Okay, don't forget your pre-lab quiz next week. First 4 pages, correct? And we're doing the inside lab. Also remember that your reading quiz will be over the last 2 sections of Chapter 6, and so it's all enzymes next week. So. In the Monday session, I will hand back your cell membrane labs, and we will go over those. One of the things I'd like to do is to show you some images of the yeast transport lab from Flask A and Flask B, and to walk you through the logic of those particular results and how you can derive and answer particular questions like you did based upon the results that you got from the lab. That's something that you will have to know to do, because on your first lab quiz there will be a similar exercise. And so make sure you understand how to do that. So we'll go over that on Monday. Any questions? Okay, last time we were talking about metabolism, as we will be for the next couple of weeks, really. And we said that, well, what is metabolism? When we say the term metabolism, what are we referring to? Go ahead. Spit it out. All the chemical reactions that take place within a cell. Okay? And remember that I said really is that's what a cell really is. It's a big chemical factory. And the result of those chemical reactions are things like the ability to store and transmit genetic information, reproduce, move. That's life. Okay? So life really is a complex set of chemical reactions. And we also said that these metabolic reactions typically occurred within pathways where something is converted into something else, which is converted into something else, etc. And in the cell's metabolism, these particular pathways are branched, they interact with one another, and it gets to be pretty complex, and so we showed you that kind of metabolic roadmap. Okay? To make sense of this, we decided, or I told you, that all these chemical reactions can kind of be lumped into 2 basic big categories. What are the 2 big categories of metabolic reactions? Anabolic and catabolic. Okay? What are the characteristics of catabolic? Catabolic reactions, do they break down or do they synthesize?
They break down.
They break down. One of the ways you can kind of remember anabolic is, you've all probably heard of anabolic steroids before, right? What do anabolic steroids do? They pump you up, okay? They increase your muscle mass. They build up. The catabolic reactions are degradative. Are the energy requiring or releasing? They're energy releasing, okay? Do the substrates tend to become oxidized or become reduced? Yeah, in these catabolic reactions, typically what you're doing is you're removing hydrogens from those particular molecules. And if you lose a hydrogen, what else are you losing? An electron, and in the process then, lose electron, oxidized, right? So something like glucose, for example, can become oxidized. And if you completely oxidize glucose and pull all of its hydrogens off, what's left? CO2, okay? So cellular respiration is a catabolic reaction. It is an energy-releasing reaction. The energy that's released is trapped in the form of ATP, and much of that energy that's released is lost in the form of waste heat, which organisms have to get rid of. The last criteria we said to define those particular reactions is whether they were spontaneous or not. Are catabolic reactions spontaneous or not spontaneous? They are spontaneous. Okay, so that's what we have to dig into a little bit more right now, is this idea of spontaneous. What does it mean? And more importantly, how can we determine whether a particular reaction is spontaneous or not? So spontaneous reactions happen on their own without an input of energy. So again, I'll use this analogy many times, but kind of like our rock sitting right at that precipice at the top of the hill. If it's given that little nudge, then it rolls down that particular hill and does so on its own without an additional input of energy. The way that we can really understand spontaneous reactions is to understand the second law of thermodynamics. Remember we said that the second law of thermodynamics could be stated in a number of different ways. The concept that I would like to use for the rest of today, and for much of the rest of the discussion of metabolism, is to discuss the second law of thermodynamics within the context of this idea of free energy. What does the second law of thermodynamics say about the behavior of matter in regards to free energy? It says that matter tends to spontaneously move from states of what to what? High free energy to low free energy. Okay? And reactions to that we are said to be spontaneous. So that kind of makes intuitive sense. I mean, this is going to happen on its own. This is going to require an input of energy to occur. So that's not too difficult to wrap your head around. So here are some examples of some things that we've talked about. Our person on the diving board, their fall through the air until they strike the water due to the influence of gravity is a spontaneous reaction. Diffusion is a spontaneous reaction. Okay? Molecules are more concentrated. The more concentrated you are, the higher free energy status you have. And they will spontaneously spread out and assume a more diffuse or spread out arrangement. This is less concentrated. So this is a movement from high free energy to low free energy. Molecules that are highly ordered can be broken into their subunits. That is a spontaneous reaction as well. This more ordered state, more ordered, lower or higher entropy? Low entropy. So this would be a low state of entropy or disorganization. This would be a higher state of entropy or disorganization. So that's a natural tendency of matter, to assume a state of greater entropy. Well, what actually determines the spontaneity of a chemical reaction? Well, if we take some kind of just generalized reaction where A and B are combining chemically to form compounds C and D. The generic term we use for A and B is substrates, or also oftentimes referred to as reactants. And then C and D represent products that are formed. Okay? And what does this particular double arrow indicate to you? That this reaction is potentially reversible, and A and B can be converted into C and D, but likewise C and D can be converted back into A and B again. Well, spontaneous reactions occur, we just told you, with a decrease in free energy. They're downhill. If a reaction isn't spontaneous, then it will proceed with an increase in free energy. So if this reaction were happen to be spontaneous, written in this direction, AB to CD, what would C plus D to AB be? If going in this direction it was spontaneous, going in the backward direction would be not spontaneous, right? Okay? Well, what we want to do is we want to define this concept of free energy. Because what does this imply? If this reaction is spontaneous going from this direction to this direction, and spontaneous reactions proceed with a decrease in free energy, then which one of these two have the higher free energy? A and B have the higher free energy, right? If the reaction written in this direction is not spontaneous. So this would be at the top of the hill, this would be at the bottom of the hill. And that reaction would proceed from from AB to CD in a downhill fashion with a decrease in free energy. Likewise, CD going back to AB would be going back uphill again. And that would not be a spontaneous reaction. Okay? So, we're defining this in terms of this concept of free energy. But what is free energy? Well, we're going to define free energy as the energy available to do work. What does that statement imply? If there is energy that is available to do work, there is also energy that must not be able to do work or available to do work. Okay? For a cell, what's a typical type of energy that the cell can produce but really can't do much work? Remember we said that heat energy was typically considered to be a waste form of energy for the cell. Under the right conditions of big temperature differences, heat energy can do a lot of work. Okay? That's how a coal-fired power plant or a nuclear power plant works, is it generates energy based upon huge differences in heat from one part of the system to the other. But can a cell set up a situation where the cell gets really, really hot in one part and not so hot in another part? No, cells are isothermal. Okay, they want to maintain a fairly constant temperature, so things don't denature. So really, for metabolism purposes, we consider heat to be kind of a wasted form of energy, basically something that isn't very available to do work. Well, this is the equation that defines free energy. G equals H minus Ts. In a cell, we're going to be really more interested in changes in free energy. And so instead of looking just at G, we're going to be looking at delta G. Anytime you see that term delta, it's referring to a change in a parameter. So we're going to be looking at free energy changes. So what does delta G represent? The maximum amount of free energy that can be harvested from a reaction. The maximum amount of energy that can be harvested that can do some meaningful work. Okay, so if I've got this reaction and written from left to right, it's spontaneous, then this has more free energy than this does. And it's going to naturally proceed from going in this direction to this direction. But not all of that energy can be harvested to do meaningful work. So delta G would represent the amount of the energy that is released, if you like, in this conversion, that the cell can use to do something, to stay alive. Well, what is delta G? What's the maximum free energy change? It's delta H minus T delta S. This represents the energy available to do work. Delta H represents the total energy of the system. If you add up all the energy in these molecules and these molecules, that would be delta H. So what is T delta S going to equal? Well, if this is the total energy, and this is the energy available to do work, what's this? This is the waste energy. This is the energy unavailable to do work. And what are two things that really affect this energy unavailable to do work? Temperature. Does anybody know what S refers to? S is a term that is used to describe entropy. So this particular unavailable energy to do work is largely driven by or determined by entropy changes in the system. Now that gets a little bit esoteric, and if that confuses you, don't get too hung up on that. So what's the take-home information you really need to get from this kind of complicated slide? So what's really going to be determining whether reactions are spontaneous or not is the free energy changes that occur. And reactions that are spontaneous are going to go from states of high free energy to states of low free energy. Reactions that aren't spontaneous are going to go from states of low free energy to states of high energy. So spontaneous downhill, not spontaneous uphill. Okay? So that's not too bad. Well, practically, how do we determine the delta G, or free energy change, of any given reaction? Well, to do that, you take the free energy of the products. Okay, so if we've got this reaction, we take the free energy of these molecules and we subtract from that the free energy of the reactants. So free energy changes is going to be products minus reactants. Okay? So if this is our A and B up here, and this is our C and D down here, what's the free energy change of this particular reaction going to be? Well, let's just throw some numbers in here. Okay? Let's say that the free energy of the product reactants is 10 units. And the free energy of the products is 1 unit. So what is going to be the delta G of this particular reaction? Is it going to be a positive value or a negative value? It's going to be a negative value, right? Because we're going to have 1 minus 10. So the delta G will be minus 9. So free energy changes can tell you by looking at the magnitude of the delta G value, and particularly whether it's a plus or minus value, whether that reaction is spontaneous or not. So negative delta G values represent reactions that are not spontaneous. Positive delta G values, negative delta G values are spontaneous. Positive delta G values are not spontaneous reactions. Okay? So if we had a reaction instead that looked like this, where this is A and B, and this is C and D, then things would look a little bit different. Okay, so if the delta G is negative, the free energy of the reactants is greater than the free energy of the products, the reaction is spontaneous. If delta G is positive, then the free energy of the reactants is less than the free energy of the products. Like this situation. Here's the reactants, here's the products. The delta G would be positive. What if the free energy of the reactants is equal to the free energy of the products? What's the delta G going to be? Zero, right? At that particular condition, we are in a state of equilibrium. And the rates of the forward and the reverse reactions are going to be equalized. So at equilibrium, delta G is equal to 0. And what does that tell you? Can any meaningful work be done by a reaction that's reached a delta G of 0 or that's reached equilibrium? No, because free energy change is 0 now. And free energy is the energy available to do work. So this is something that you really need to wrap your head around, because we're going to be talking about delta G's of various reactions as we go through cellular respiration and photosynthesis. Okay, so let's put this together. Okay, so in all these particular situations of the person sitting on top of the slide and sliding down, or molecules diffusing, or complex ordered molecules being converted into less ordered molecules. In all these particular situations, these represent states of high free energy. These represent states of low free energy. These are spontaneous processes. How do we know that? Because the free energy of the products down here is less than the free energy of the reactants. And if we calculate delta G's for these, the If we take this and subtract this away, we'll get a negative value. So all of these would have negative delta G's, or spontaneous reactions. Let's take a look at this graphically, okay? So on this particular plot, what do we have? As you go up this particular axis right here, the y-axis, we've got increasing free energy. As we go along the x-axis over here, we have what we call progressive reaction. Okay, so if this is our reaction, right over here, pretty much all we have are molecules of A and B. As we move in this direction, some of A and B is going to be converted into C and D. So this kind of represents the reaction going in this particular direction. Okay? So, free energy of the reactants is big. The free energy of the products is less than that. So this represents a downhill spontaneous reaction. It will be energy releasing. How much energy can be released? The difference between this energy and this energy. And that represents the delta G for the reaction. And that value would be negative because remember, products minus reactants. Something small taking something bigger away from it. Here's another type of reaction. Here, the free energy of the reactants is less than that of the products. So this will not be a spontaneous reaction. This will require an input of a reaction to drive the formation of products. The delta G represents the difference between the free energy of the products and the free energy of the reactants. And because this is a bigger value than this, then the delta G values will be positive. So this would represent the amount of energy required to force the reaction toward completion. Now, a couple of new terms that are introduced in this slide. Exergonic and endergonic. The first reaction that we talked about is characterized as being an exergonic reaction. Exergonic reactions are those reactions where energy is released as they spontaneously occur. Exergonic reactions are characterized by having a delta G of less than 0. They have a negative delta G. What if the form of the energy that's released in these reactions is heat energy? Do you know what we call those reactions then? Have you heard the term exothermic before? Okay, heat-releasing reaction. Sometimes you mix two chemicals together in a test tube, and the test tube gets warm. That's because the reaction is exothermic. It's energy releasing. In this particular case, the energy that's released is heat. So exergonic is more of a kind of a general term describing energy release. Exothermic refers to the release of heat energy specifically. Endergonic reactions have a positive delta G because they require inputs of energy. The free energy of the reactants is less than that of the products. Sometimes if you mix two things together in a test tube, the test tube actually gets cooler. And you can feel that. That would be an endothermic reaction. Okay? So exergonic and endergonic. Exergonic are spontaneous. Endergonic are not spontaneous. Okay, we already talked about that. So, here's a good review slide, hint, hint, for your exam. Because these are terms that you're really going to have to be able to distinguish between. So, if we look at exergonic first, what characterizes them? Negative delta G values. So what does that tell you? These reactions are spontaneous. They tend to occur naturally on their own. They're downhill. They're going from states of high free energy to states of low free energy. States of high free energy are unstable. States of low free energy are stable. Unstable states, or states of high free energy, tend to be states of low entropy. They tend to move to states of high entropy. So what does this negative delta G represent? The maximum amount of work that the reaction can produce. Why is it negative? Because the free energy of the reactants is greater than the free energy of the products. So you see how all these things kind of tie together? And it's important for you to understand the relationship between those particular terms, but particularly this relationship between exergonic, spontaneous, and free energy levels, because that's something that we're going to apply over and over again as we talk about cellular metabolism. Endergonic reaction is pretty much just the opposite. So if you understand this column, you understand this column. Okay, so now let's talk a little bit about metabolic reactions and equilibrium. Would it be a problem for the cell if all of its metabolic reactions, especially the catabolic ones, were allowed to reach equilibrium? Okay, remember what catabolic reactions do. They're degradative, they produce energy, they're exergonic. And the cell needs energy, right? But what would happen if those reactions were allowed to reach equilibrium? The rate of the forward reaction was allowed to balance by the rate of the reverse reaction. Then could that reaction do any meaningful work? No, because our delta G would be 0. The free energy change would be 0. Well, fortunately, the cell usually doesn't allow metabolism to reach metabolic equilibrium. It plays some tricks to try to ensure that these reactions don't reach equilibrium. One of the things that allows that to work is that, remember, metabolic reactions tend to be multi-step reactions, where one thing is converted to another, is converted to another. So the product of one reaction, this reaction, can then be the substrate of a subsequent reaction. Cells are constantly trying to keep their reactions from reaching equilibrium. We can say that the cells kind of practice non-equilibrium metabolism. The only reason that they are able to do this is because they are open systems. Remember what an open system involves? If you're an open system, what can you do? You can exchange energy and matter with your environment, right? You can bring things in, you can get rid of things. Let's take a look at a simple example to illustrate this. And we'll look at this simple hydroelectric system. This hydroelectric system really is going to involve basically 2 canisters of liquid, a little wheel or turbine in this chamber that connects the 2 canisters, and then that turbine is hooked by some wires to a light bulb. Okay? In this particular system right here, the light bulb will light, right? Because the water level is higher on this side than this side. The free energy is higher over here than over here. And this reaction will proceed spontaneously from high free energy to low free energy. The free energy difference will be negative, because we take this minus this to calculate free energy difference. And that energy that is released as the water falls will be used to turn the turbine and generate electricity. Okay? But what's eventually going to happen? The water levels will equalize, right? And now there still can be some molecular movement. Some water molecules can still move over here, but others will be moving back. So the free energy change will be 0. Our light's going to go out. Okay? There is no free energy that is being harvested now to power this particular light bulb. Why? Because this is a closed system that has been allowed to reach equilibrium. Here's an example of an open system. This particular system can be used to continually generate electricity if there is a constant input of water into this container and a constant output of water from this container. So we maintain our water level differences, we maintain our free energy difference, and that can be tapped to light the bulb. So this represents an open system. This is what it sounds like, okay? Well, here is a multi-step hydroelectric system involving several containers. Each one of these individual steps represents an opportunity to generate some electricity as long as we maintain free energy differences between those particular containers. But this only works if it's an open system. When we constantly bring things in, like food, and get rid of things here, waste products. This is analogous to what's happening in a cell's metabolic reactions. Why isn't this reaction reaching equilibrium? Because this container then empties into this container. This isn't reaching equilibrium because this container is emptying into this container. And so we have a stair step of free energy differences, and each point in that sequence allows energy to be harvested in the form of light in this case, trapping some of that free energy that's released as water goes from high free energy to low free energy. So as long as we constantly remove C and bring in more A, this particular reaction will not reach equilibrium. And therefore delta G won't be zero. And this can keep generating energy, if you like, to use to provide for cellular needs. So one of the reasons that metabolism is not equilibrium is the fact that our metabolic reactions are set up in these stepwise sequences where the product of one reaction becomes the substrate of a subsequent reaction. One of the things we're going to be talking about is the fact that in cellular metabolism, endergonic reactions are commonly coupled with exergonic reactions. Well, that makes sense if you think about it. Energy requiring reactions are tied together with energy releasing reactions. Okay? What's an example? Well, energy from the breakdown of glucose, for example, by catabolism is an exergonic process, energy releasing, and some of that energy can be used to synthesize or to make ATP. Okay? ATP can then be broken down, and the energy release can be used to power cellular work. But then we need more energy from catabolism to resynthesize ATP again. So this particular exergonic process is coupled to this endergonic process of making more ATP. Here's another example. This particular reaction of the production of a compound called glutamine from glutamic acid and ammonia has a delta G of plus 3.4 kilocalories per mole. So is that reaction exergonic or endergonic? Endergonic, right? Positive value endergonic. So this has higher free energy than this does, okay? It's uphill. But the cell needs to do this, okay? This is an important part of cellular metabolism. ATP hydrolysis is exergonic, minus 7.3 kilocalories per mole. If we couple these two reactions together, if they occur in partnership with one another, and that typically involves the phosphate group released from ATP hydrolysis phosphorylating one of these compounds, then if you take these two reactions together, the overall delta G of this combined reaction is the sum of the delta G's of the individual reaction. So this particular delta G of this coupled reaction of glutamine synthesis coupled to ATP hydrolysis is negative 3.9 kilocalories per mole. So overall, this is spontaneous. The uphill part of the reaction is driven by a downhill part of the reaction. Okay? What if the delta G for ATP hydrolysis was minus 2.3 kilocalories per mole? Then could that effectively be coupled to glutamine synthesis? No. Because plus 3.4 and minus 2.3 would still give us a positive value. So overall, this reaction would still be endergonic. So it's kind of fortunate for cellular metabolism that ATP hydrolysis has a fairly large negative delta G value. And therefore, that can be coupled to reactions that have a reasonable requirement for an input of energy. So we're going to see a lot of that in cellular respiration as we talk about metabolism. Okay, so that's the dreaded thermodynamics. So we're going to apply some of that now as we talk about cellular metabolism in more detail. So here's a chemical reaction. Here we've got sucrose. Sucrose is what kind of a molecule? How do we characterize this molecule that has these two monosaccharides? It's a disaccharide, right? And these two monosaccharides are, you know what they are? This is glucose and this is fructose. Okay? And if you bond them together, the disaccharide is sucrose. Chemical reaction can occur, which is a hydrolysis reaction, meaning this particular bond will be split. Some of the hydrogens from water will go over on this sugar. Some of the hydroxyls from water will go over on this sugar, giving us two monosaccharides. Okay? Sucrose hydrolysis overall has a negative delta G value. So spontaneous or not spontaneous? It is spontaneous. Okay, it's an exergonic reaction. So what does that mean? That means if I put a pile of table sugar out here on the bench and let it sit, it will on its own eventually break down into glucose and fructose. Okay? How long is that going to take? A long, long time. Okay, that sugar that sits in your sugar bowl doesn't overnight turn into glucose and fructose. So when we get a delta G value for a reaction, which is determined by what? How much free energy this has compared to how much free energy these have. Okay, this is higher free energy than this is. That delta G value will tell you whether it's go or no go on its own. But delta G value doesn't tell you anything about how fast that happens. It doesn't tell you about rates of reaction. As it turns out, the rate of sucrose hydrolysis is really, really slow. What is the rate? If you're in your car, what is your rate of travel? How do we express it? Miles per hour, right? Rates are just some kind of an amount over some period of time. So the rate of conversion of sucrose into glucose and fructose is a very slow rate, even though the reaction is spontaneous. Same thing is to be true with glucose, okay? The conversion of glucose in the presence of oxygen being converted to carbon dioxide and water, cellular respiration, overall has a negative delta G value. But its rate is also really, really slow. Okay? We've got bottles of glucose sitting around in our storage rooms and stuff that have been there for years, and they're still glucose. Well, this reaction, which also produces ATP, is really important to the cell, right? And remember that we said if you're a brain cell, if you're deprived of oxygen, then you can't do this, and therefore your brain cells run out of ATP. And how long does it take brain cells to die? A few minutes, okay? If I just take a pile of glucose sitting in the air and leave it out on the tabletop, it's going to take forever for that to decompose into carbon dioxide and water. So as far as cellular metabolism is concerned, really slow rates of spontaneous reactions can be really problematic, right? And yet, this particular reaction can occur under the proper conditions like that, pretty much. Why? What information is given to you up there that tells you why that rate of reaction might be very high under certain circumstances? It's got an enzyme, right? Sucrase. And typically, common name of enzymes end in ASE. That's your tip, that it's an enzyme. And typically the name is derived from what's one of the substrates of that particular reaction that the enzyme catalyzes. So, tell me what you know about enzymes. Okay? All enzymes are proteins, right? Sure. That's what we used to think. Now we have to qualify it and say most enzymes are proteins. There actually are some RNA molecules that have enzymatic activity. Those RNA molecules that have enzymatic activity are called ribozymes. And we'll come back and encounter a few of those later on. But practically speaking, most enzymes are proteins. Okay? What else do you know about enzymes? What do? They catalyze reactions. So what's a catalyst? What makes something a catalyst, or what can catalysts do? They can speed up a reaction. They can increase the rate of the reaction. How fast it occurs. How fast sucrose is broken down. Are all catalysts enzymes? Can you think of another kind of compound that sometimes is a catalyst? There are some metallic compounds which speed up the rate of reaction. Some of them are in your car in a pollution control device called a catalytic converter. And they actually speed up the rate of a reaction that removes pollutants from your exhaust. Metals like platinum can be catalysts. Sometimes acidic compounds can be catalysts. There's a phenomenon called acid catalysis. But the cell doesn't use these kind of catalysts to speed up its metabolism. The cell uses protein and, in some cases, RNA molecules. So they use organic catalysts as opposed to inorganic. What's another kind of characteristic feature of a catalyst? You typically need to add a lot of catalyst to the system. No, you typically don't. Usually catalysts are effective in pretty small amounts. How come? I mean, if you throw it in, or throw a catalyst in, then don't you have to throw some more in? Why not? I mean, aren't they consumed in the reaction? No. Catalysts are constantly regenerated during the reaction. They're not used up. They're not consumed. They're recycled, if you like. Some enzymes require a molecule that is kind of a helper. If these are also organic molecules, these particular helpers are called coenzymes. Okay, so they're kind of a partner that work with the enzymes. Does anybody know an example of a coenzyme that's really important in cellular metabolism? Have you ever heard of NAD before? You're going to get sick of hearing about NAD when we talk about cellular respiration. If the coenzyme isn't present in the proper form, then the enzyme won't work properly. Now, what does that imply? Well, that actually implies that there's more than one form of this coenzyme. And we'll talk about this later on, but another form of this coenzyme is a form of coenzyme called NADH. This is the reduced form, and this is the oxidized form. And so enzymes can be really, really picky about what partners they pair up with. And in cellular respiration, this is the form of the coenzyme that's really, really important if the enzyme is going to work. So in cellular metabolism, there can be really an issue. Is there enough oxidized NAD around to partner with the enzymes that are needed to make cellular respiration work? So we're going to come back and talk about that one in quite a bit more detail. But for right now, it's important to understand that some enzymes require this helper molecule to be effective. So this is probably a good place to then pick up on Tuesday. Is we probably should be able to finish up with enzymes on Tuesday. So most of you for lab will have been through this particular material, but all of you should have read this in preparation and for the quiz on Monday. Okay? Have a good weekend. Arthur? No,